Martina and I are here today talking about eutrophication, which is the enrichment of an ecosystem, a lake or a stream, for example, with chemical nutrients. But what kind of chemical nutrients, Martina?
Well, I think it's mostly nitrogen and phosphorus, and they are generated like from runoffs of fertilizer from lawns, gardens, and fields.
Okay. Well, one of the reasons that we wanted to study eutrophication in the lab is because there have been some incidents of eutrophication here in Idaho that have been a problem. You want to tell us about one?
Yeah, there's actually an example very close to home, Fernand Lake in the Idaho Panhandle, and it was characterized by foul-smelling water, blue-green algae that might be producing toxins that are harmful to all animals, including humans.
Wow, so that does become a problem. Does this happen? I noticed that this happened in the summer. Is that typically when we have these problems?
Yeah, because one thing is the increase in sun energy and then also temperature.
Oh, perfect. Bad conditions, great for us, but bad for eutrophication.
Okay, well, this diagram shows actually very well what's happening, at least in some detail. So nutrients, the nitrate and the phosphate, are usually limiting because the plants have plenty of CO2. But now, adding nitrate and phosphate, they need this to make more protein and more DNA. And if they have that, then they can make more of themselves. So they grow really rapidly, because they have all the CO2 they need, they have all the sun they need, and now they also have all the nitrate and phosphate that they need.
So really this is only autotrophs, only organisms that can fix carbon dioxide can do this. Okay.
So then it looks like then they float up onto the surface.
Okay, so that's what's going on here. And I do want to point out in this particular diagram, it says algal blooms, algae blooms, but in fact most of these are cyanobacteria, aren't they?
I think so.
Yeah, it's not really algae, it's cyanobacteria. And as you know, those are also photosynthetic. And they look a lot like algae, but they give algae a bad name because it's really the cyanobacteria. They are the ones that can float up to the surface like this because they make gas vesicles. But then what happens?
So then, if you have a thick green layer on top, then the sun can't go through. And they also are exposed to too much sunlight. So then they start dying off because now the sun actually causes them to kind of have a sunburn. They die.
And this is when it looks like oxygen gets depleted and plants die?
Plants die. Well, the plants die because they can't get sun.
Right.
The algae die because they get too much sun.
Okay.
Everybody falls down to the bottom. Now, bacteria in general, they need nutrient-rich environments, so they have
those are the decomposers down here?
Right, and they can be aerobic or anaerobic. If they are anaerobic, they have no problem. They can grow whenever. But if they're aerobic, they initially grow because now they have a lot more carbon. But then they start dying as well because since the plants can't make more oxygen.
So they're consuming the oxygen, and pretty soon our fish die, right?
Yeah.
All right.
And then the fish die too.
So now we have this stage that we get to, which is pretty awful, the death of the ecosystem, until this can somehow recover, which doesn't always recover, does it?
No, I don't think so, because, well, if you can sequester all of this biomaterial down into the sediment, and that sometimes happens in the winter, then the next year you might have more fresher water.
Yeah, but I know that I was in Minnesota this summer, and one of the things that they had as their headline news was that a lot of the lakes that have gone eutrophic are not recovering this time. They're recovering much more slowly because these lakes are going eutrophic for longer periods of time.
Right, and then they have dissolve that organic material, and it starts out with low oxygen load.
Okay, good, wow. Well, this is a nice cartoon showing us really the contrast between healthy system on the left and a eutrophic system on the right. And clearly one of the key differences is that we've got minimal input of nutrients here, and that allows for balanced growth of algae and cyanobacteria. We've got pretty healthy fish because they have a lot of oxygen, and of course the healthy grasses that are normally lining the bottoms of these lakes and rivers because the sunlight can actually get through to them. But let's contrast what's happening here.
Isn't it amazing that just a little bit more input of nutrients like phosphate and nitrate causes everything to just spin out of whack? So basically it causes all the problems that we encounter with a eutrophic system.
Right, so no oxygen, and of course this is exacerbated by the temperatures during the summer because that's going to drive off the oxygen, right?
Even more.
Yeah, and the algae, when they bloom, they, are, of course, in response to these extra nutrients, start to die off and cause all of this other decomposition that you mentioned earlier. I want to mention one more thing about the algae and cyanobacteria in particular, is that they can produce toxins. We don't know all of the conditions that favor toxin production, but the toxins can also kill a lot of animals in these environments if toxins get produced. Well, one reason we thought this would be a good experiment for this project to look at eutrophication is because there's a lot of possibility for interdisciplinary work here. And I think in this diagram, we're looking at contributions from biology in orange, and yellow is probably from chemistry, I think.
Chemistry, yeah, it looks like chemistry is a good partner there. And then also we have the microbiologists.
So what are each of those groups going to do?
So for chemistry, they can look at what kind of chemicals are, or what are the levels of these nutrients, the phosphate and the nitrates. They can also look at are there some toxic chemicals produced.
Okay, so maybe the microbiology students can try to determine what kind of microbes are there, and since cyanobacteria are bacteria, maybe the microbiologists can look at that and actually try to maybe even purify, characterize, identify the microbe.
Yeah, and the same thing is then for the biologists, except they would focus on the protists, like the cells. I almost said real cells. Well, I think the cells that have a nucleus. And also the photosynthetic ones, and they may even get, well, actually, I was just thinking the microbiologists can also look at those other microbes that grow because of increased organic matter available.
And hopefully if they can identify some of those, they can actually get a microbe that's producing a toxin and hand that off to the students in chemistry for some characterization.
Yeah.
But of course, we're curious about what happens to the fish. Do they die because there's no oxygen, or is it because of the toxin, or is it because of the nitrate? Do we know?
I don't think so. I'm not really aware of how nitrate affects fish.
Okay, I know that's kind of an open question. So hopefully the students will be able to ask some of those questions and really pin down what's going on in some of these environments.
I think that's a good idea. So.